Good morning, everyone. Myself, suman purbia from group 316. Here is my topic protein biosynthesis. Protein biosynthesis or a protein synthesis, it is a core biological process that occurring inside the cell. The template principle of a protein biosynthesis is a fundamental to the molecular biology and the molecular biophysics. We know that the information contained in a gene, that is in the DNA, which is transferred to the mRNA during the process known as a transcription. The protein synthesis takes place exactly on a mRNA. This is called the central dogma of the molecular biology, that states that the genetic information flow from the DNA to the RNA, RNA to the protein. Let's see in the diagram. The DNA which is present in the nucleus, that is present inside the cell, this DNA form the mRNA by the process known as transcription. This mRNA comes out in the cytoplasm and the ribosome bind to the mRNA which form the protein. This process is known as a translation. We have the genetic information can never be transferred from the protein to the nucleic acid, that is the mRNA and the DNA. We have an exception of this, that is the reverse transcription. It is a process where the transmission of a genetic information flow from the RNA to the DNA. In the 1970s, two scientists, Thamin and Baltimore, discovered this process where the RNA prime or the RNA of a prokaryote will transfer the information to the DNA. This DNA further form the RNA which form then protein. This is a reverse transcription. For the protein synthesis to accomplish, there is a starting material that is known as an amino acid. The amino acids are present in the cell in the free state. The direct polycondensation of the amino acid is an endergonic process because it increases the free energy during the formation of a peptide bond. In the cell, the polycondensation of amino acid is coupled with the exogonic process of dephosphorylation of the ATP. Let's see in the reaction. This is an amino acid which bind to the ATP in the presence of enzyme known as amino acyl tRNA synthetase, which form the amino adenylate and pyrophosphate is released. Now, protein biosynthesis occur at the ribosome. The ribosomes are nuclear particle composed of protein and rRNA. The ribosomes provide the appropriate interaction between the mRNA and the tRNA, which carry the amino acid and the polycondensation of the amino acid to form a polypeptide chain. The ribosome plays the role of a catalyst, which is similar to the action of the enzymes. Here we have a diagram. This is a gene which by the process of a transcription in the nucleus it form the mRNA. This mRNA comes out in the cytoplasm and where the ribosome is bind and the tRNA comes, the polypeptide chain is formed that then further will form the protein. This process is known as a translation. The ribosome attached at the 5' end, that is here 5' end of the mRNA, which codes the end terminus of the polypeptide chain. The ribosome moves along the mRNA chain, reading the codon to codon after codon. This is a ribosome. Here we can say that this is an mRNA. This ribosome, here the mRNA will come, tRNA will come and it's bind to the mRNA. The tRNA and T codon bind to the codon of the mRNA and the polypeptide chains start forming. The synthesis of a protein chains on a polysomes is known as a translation. The chain or a several chain gives rise to the biological function protein molecule. Transcription. Transcription or a DNA synthesis is a process of creating an equivalent RNA copy of a sequence of a DNA. Both RNA and a DNA are the nucleic acids which use a base pair of a nucleotide as a complementary language that can be converted back and forth from DNA to the RNA in the presence of enzymes. DNA will form the RNA. This RNA will form the protein. We know that is known as a central dogma, and this process is known as a transcription, and this process is known as a translation. During transcription, a DNA sequence read by the RNA polymerase, which produces a complementary anti-parallel RNA strand. Transcription is the first step leading to a gene expression. The stage of a DNA transcribed into a RNA molecule is called as a transcription unit and encode at least one gene. Transcription is divided into three states. First is the initiation, second is the elongation, and third is the termination. Let's see in the diagram. Initiation in the bacteria, the transcription begins with the binding of a RNA polymerase, which having a sigma factor. Along with this, it bind to a promoter of the DNA helix, so that the initiation will start. And then when the initiation start, after that after initiation, the second process of the transcription start, that is the elongation. In an elongation process, the one strand of the DNA of the template strand is used as a template for the synthesis of the RNA. This RNA strand is continuously growing, and this RNA polymerase, when this RNA polymerase is reached to the terminator region of the DNA helix, then the third process is started, that is the termination process. In this termination process, the Rho factor bind to the RNA polymerase, and the synthesis of the nascent RNA is stopped. Now, transcription in eukaryotes. Transcription in eukaryotes are more complicated than in the prokaryotes. In eukaryotes, we have three different type of RNA polymerase, that is the RNA polymerase 1, 2 and 3. We have three different functions of these RNA polymerase, and it also requires many transcription factors and proteins. This transcription initiated need the promoter and the upstream regulatory region. Here the enhancers and silencers of are DNA sequences that regulate the rate of the initiation of the transcription by the RNA polymerase second. Let's see the diagram. This is in a DNA helix. This is an RNA polymerase. The process is occurring same in the eukaryotes and the prokaryotes, but there is some complexities that is occurring in the eukaryote. That is, this RNA polymerase produce a RNA that is not mRNA, that produce a heterogeneous RNA, where it means it produces exons and the introns that are they both are the non-functional. So we have to do some additional process. First is the capping. We add a methylene guanosine triphosphate at the 5' end. Then we add a poly A tail at the 3' end, and the RNA splicing will occur. Here the removal of the introns will occur, and exons are further joined by each other, and the formation of a new mRNA will occur. This process is known as a transcription in the eukaryote. Now let's start another topic, that is the reverse transcription. In the reverse transcription, the RNA is forming the DNA by the process known as a reverse transcription, and the enzyme is used is the reverse transcriptase. In 1970, Tamin and Baltimore discovered this process and this enzyme, reverse transcriptase. This reverse transcriptase is an RNA dependent DNA polymerase. The process of a reverse transcription, first is the annealing of a primer to the RNA transcription or to the poly A tail. Second is a reverse transcription by the reverse transcriptase enzyme. Third is the removal of the mismatched by RNase H. Fourth is the repairing of this gap by the DNA polymerase 1, that is also known as the form. This gaps is formed and this fragments is known as the kino fragments. Then the fifth step is coming, that is the downstream processing of the complementary DNA. Let's see in the diagram. This is our DNA strand, and this is the primer. The first step is the annealing of the primer. Then in the second step, the reverse transcriptase will form these RNA. Then after that, the RNase H will come, and the mismatched remove the mismatched RNA. So if this mismatched RNA removed, the fragments will form. Then after the RNA polymerase 1, they repair these gaps, and the perfect complementary DNA will form. So this is all about today's topic. Thank you.